So, a very good morning to everyone present here. I, Dr. Rajkumar Arya, Associate Professor in Department of Chemical Engineering, NIT Jalandhar, the course coordinator of this international short-term course on current and future scope of surface coatings. The patron of this particular course is Dr. Lalit Kumar Avasthi, the Director of NIT Jalandhar. And convener of the course is Dr. Sangeeta Garg, the head of the department. So today is fifth day of our this short-term course, and the speaker of this morning is Dr. Ganpat Dahe, who, who who will talk on application of biocompatible polymers on hollow fiber Lumen. So before starting off his talk, let me introduce Dr. Dahe to all of you present. So we have amidst us Dr. Ganpat J. Dahe, who is a coating surface manager with Hydromer Incorporation USA in Concord. Dr. Dahe is a material, product, and process development expert with industrial experience in polymer extrusion of fibers and films of biomedical and industrial coatings, structure property optimization, and material characterizations. Prior to joining his current organization, he was associated with Ledell Performance Materials as a senior material scientist, where he led the development of four key polymeric sealing products to retain market competitiveness. He was also instrumental in developing wire-coated polyacrylate-based press in place sealing product. Dr. Dahe also worked with National Energy Technology Laboratory as a research engineer and Los Alamos National Laboratory as a postdoctoral associate. He was also associated as a graduate research associate with IIT Bombay. On the educational front, Dr. Ganpat Dahe is a PhD in chemical engineering from IIT Bombay and Bachelor of Science in Petrochemical Engineering from University of Pune. Dr. Dahe is the recipient of the several awards like LAAP Awards for Outstanding MOB and LANL and CINT User Proposal Grant at Los Alamos National Laboratory, USA. He has two patents to his credit and has been affiliated by Polymeric Society ACL, the rubber the division. So before we start the session, I would like to mention also Dr. Dahe and me did a PhD from IIT Bombay. He is my classmate. So with this brief introduction, now I will ask Dr. Dahe to start the session.
Thank you, Professor Arya, for introduction, and thank you for the opportunity to discuss the work I have conducted in polymers and coatings. So, can I share my screen? How? Let me
Yes,
see.
yes, sure.
Okay, can you see my screen now?
Yeah, it's coming.
So today, I'm going to present on the application of biocompatible polymer to hollow fiber lumen.
Dr. Ganpat, can you hide this bottom bar there? The other one bottom bar that hide presentation.
Yes.
Yeah,
Can you?
yeah, thank you.
Okay, sounds
Yeah.
good. So, this is the outline of my presentation. First, I will introduce you to the hollow fiber membrane, how they are prepared, what's the application, some of the motivation to applying the biocompatible polymer to hollow fiber membrane, so objective of the research. And here I'm also going to discuss about some of the characterization technique that you never heard before. So I'm going to give some brief introduction about that. And here are two studies that I'm going to present. One is the development of biomaterial, that is the polysulfone vitamin E polysulfone composite membrane. Another one is in situ coating of like MPC. It's a biocompatible polymer. It's a biomimetic polymer basically on hollow fiber lumen. And then finally, like some of the in vitro evaluations study of those biomaterial in some animal studies I'm going to present. And finally the conclusion. So the hollow fiber membranes are quite thin membranes with ID of like 200 micrometer and the wall thickness around like 20, 35 to 50 micrometer. And there are like these membranes are widely used in like hemodialysis application, blood oxygenator, and some of the plasmapheresis or some like heart lung machines. So these are like the very life-saving devices. And so just I want to give some brief introduction. Like here, like the main objective of this study was to make a hollow fiber membrane for hemodialysis application. So I want to give you some introduction about what is hemodialysis. So hemodialysis is a method of removing the metabolic waste product from the blood, and these waste products include urea, creatinine, uric acid, some inorganic phosphate, and these are the kind of the toxins in the blood that need to be removed. And usually this is performed by a kidney, but this is a kind of uh, a substitute treatment for like end stage renal disease. So it's also important like to keep some of the blood protein in from filtering out. So such as albumin, immunoglobulin, those protein need to be there in the blood. So it has to like it basically filter the low molecular weight toxins from the blood. And parallelly there is another solution. It is passed on other side of the membrane, which they call dialysate. It's nothing but the it's nothing but the electrolyte solution, which has very similar the composition that of blood plasma. So that dialysate removes like take out the toxin from the blood via diffusion process. So this is a typical the diagram, like a schematic diagram, the clinical process of hemodialysis. So the blood is taken from the vein, it goes through uh, the blood pump, it pumps the blood through the lumen of the hollow fiber, and then simultaneously there is another solution that pass outside of the hollow fiber in the cell side, in the cell side, and it's a kind of like a counter diffusion, like it's a counter current process where the blood flow from one side to other side, and the dialysis flow from others like one side like other side to this side. So and that's how like it increases the diffusion faster, and that way it helps to remove the toxins very quickly. So this is the typical like hemodialysis cartridge. It usually has 10,000 fibers packed inside this membrane. So why the one of the question is that like there are like these hollow fibers being used in since long time, but there are a lot of issues associated with these fibers. And one of the like any surface that comes in contact with the blood, the first thing is that there is a deposition of the blood proteins, the blood cells, and this deposition like this blood cells they sense the foreign matter, and it led to some unwanted reactions in the blood. So for example, the the blood platelet, when they get deposited on the surface, it deforms, it led to the it deforms, sometime it ruptures, and it led to like activation of some, like contact activation is a kind of one type of process that happens during the coagulation. That's the initial process. And so there are various like inflammatory reaction triggers. Also, like some of the protein, like the beta 2 microglobulin, this is a one of the toxin that need to be removed during the dialysis, and that therefore like one has to use very like high flux dialysis membrane with a very narrow pore size. And all, if you consider the collective like effect of this, these things led to the systematic changes to the blood and organ, and it increased the mortality. So there is a need for the biocompatible surface uh, where where like one can minimize or no deposition of like a blood protein or the blood cells on the membrane. So that's the reason the objective of this project was to create a biocompatible surface that will reduce the protein fouling, the adhesion of the cells. So these are like some of like these are the desirable characteristics of hemodialysis membrane. So one is a like a biocompatibility where there is no attachment of the cells to the blood contacting surface. There should be like a there it should produce less reactive oxygen species, they called ROS, and the protein adsorption. It should have a less platelet adhesion and its activation. Finally, the complement activation is a kind of uh, immunological the process that led to that activate the immune system. The next characteristic that fiber should have, it should have very high flux, so it can remove the beta 2 microglobulin, and the molecular weight should be in between 20 to 30,000. So that way it will not remove the proteins such as albumin from the blood, which has the molecular weight around like 57,000. So and another criteria of that the membrane should be the pore size should be narrow. Next, the microstructure. The microstructure of the membrane plays important role for adhesion. Like, and if you have very rough microstructure, there is more fouling, more stuff get attached. So it needs very smooth surface, and also like this inner side of the membrane consists of the selective layer that need to be uh, formed and have a nice smooth surface. If you have any questions, you can interrupt me in between if you don't understand. But like uh, I have no problem like giving you the answering those questions. So can you hear me? Hello.
Yes, yes, we can hear you. You can go ahead. Yeah, yeah, we
Okay,
can
okay,
hear you.
okay. No, sometime I have to make sure, like,
Yeah.
I will keep talking and I want, or like maybe I realize later that, oh, I'm disconnected or something happened.
No, no.
That's the reason.
Don't worry, I would inform you actually in that
Okay,
case.
okay, okay, yeah. Just call me on my phone in case, like, such thing happen, because probably, yeah, I hope that will not happen, but yeah. So,
Sure, sure.
so the overall objective of this study was to develop the hollow fiber membrane with improved biocompatibility and separation performance for hemodialysis application. So some of the like I really wanted to like discuss these things. This is a very simple setup that I have created during my PhD. So in the there are like a two tanks, and basically this hollow fiber is nothing but extrusion process. In the first tank, we like called the dope solution tank, where polymer solution is prepared, like a polymer solutions around like 15 to 30% polymer solids are prepared, and the minimal viscosity of this solution should be like 10,000 centipoise. Then another solution they called a coagulant, also called as non-solvent, and the both solution usually pass through the spinneret. And the spinneret is one of the key to produce this hollow fiber membrane. And the spinneret has a annulus and the bore, and they call like a tubine orifice spinneret. Through the bore, the coagulant flow, and through the annulus, this polymer solution flows extruded. So all this like setup was temperature control, so one can get a consistent result irrespective of the what temperature is outside or in the room. And at this spinneret, like as soon as the polymer solution and solvent get contacted each other just below this spinneret, the precipitation or phase separation process start. And in this case, there is a diffusion of non-solvent in the polymer solution, and the solvent in the non-solvent in the non-solvent. So, and this led to like during this phase separation, it's led to like form the polymer rich and polymer poor phase, and the polymer rich poor phase led to the pore formation, while the polymer rich phase leads to the solid structure formation, like polymer matrix formation. And this is how the pores being created in the hollow fiber. This is just like I'm telling you very simple language. And then afterward, like this fiber like pass to the air gap. This air gap is responsible for outside structure determination, outside of the hollow fiber structure determination. So it depends upon the moisture, or some you can also use a third uh, second coagulant as a third orifice, and that way like you can also manipulate the structure of outside of this hollow fiber. And finally, this fiber is immersed in water, where the phase separation happens from outside to inside. And here this length between the spinneret and the water level or the coagulant level of this, that the liquid level of this coagulant tank also determine uh, the structure on outside of the hollow fiber. And you can vary the structure by varying the air length or the air gap, and this fibers pass through this coagulation tank. Then and in coagulation tank, one has to make sure that all the fibers is completely precipitated. And in later on, it passes to the next tank is called rinse tank, where the residual solvents are removed, and later on it goes to uh, the the fiber winder, the roll. So this is the typical like a schematic for hollow fiber making process. This was the setup that was developed. So here we use the nitrogen cylinder to make sure that it provide the initial, because this is a really viscous solution, and it need like that initial pressure, so that way the solution can be pushed to the gear pump, and it provide the initial suction pressure, so it will be continuously extruded or continuously flow through the spinneret. Also provide some blanketing, like it also protect the coating solution getting from the moistures and other stuff. So this is the typical hollow fiber membranes that was produced from the same setup. And these are the some ACM micrograph of the hollow fiber. You can see some of this is nothing but called macrovites. It is formed during when the precipitation is really fast, and when the mass transport happen through the more convective mass transport rather than the diffusion. And this microstructure, these structures are formed, uh, and and this is very typical, like they call like a fingerprint, like a finger like a cavities called macrovites. So here, like one of the characteristic technique that mostly the people use is the pure water permeability. It's a very simple technique, and what you have to do actually, you have to find the water permeability per meter square per of the pressure that you apply uh, through the membrane. And this is a typical formula like the people use to calculate the water permeability. The next another important characteristic of the hollow fiber membrane is to find the solute rejection. So this is like in this case, the different molecular of like a polysaccharide, like mostly people use a polysaccharide or some polyethylene glycol, or some also they use the protein solution to find the rejection. So in general, what they do actually, they create the mixture of the different polysaccharide, like such as dextran, for like from 5,000 molecular weight to all the way like a few 100,000 molecular weight. They make a mixture, they pass through that mixture through the lumen of the hollow fiber, and they measure the concentration of the dextran from permeate as well as the retentate side, and based on that, the one generate the rejection curve. And this is really important to find the molecular weight cutoff of the membrane. The molecular weight cutoff is nothing but the molecular weight corresponding to 90% rejection, and which is a very typical characteristic of the like a characterization of the membrane. So this is all about the how the membranes are produced, their characterization technique that we discussed so far. And now I'm going to talk about uh, the making the biocompatible hollow fiber membrane. And here, like what we did actually, we use a polysulfone and vitamin E TPGS, the polymer to make this membrane. We use in this case the membrane forming system consists of polysulfone, vitamin E, TPGS, and both these materials are dissolved in N-methyl pyrrolidine, which is a very good solvent. Like NMP is a very good solvent for polysulfone. And here the concentration of the vitamin E varied from 5 to 20%. And this research has been published in Biomaterial. You can also refer this article. You can find more details of this research in that article. So the question is why we use a vitamin E TPGS as additives. So vitamin E TPGS is a water soluble like amphiphilic surfactant polymer. It has a vitamin T and polyethylene glycol, and in the vitamin E, like that section acts as a head, and the polyethylene glycol section as a tail. And this surfactant like characteristic like provide plays a good role in forming the skin of the membrane, which is called the selective layer of the membrane. And in this case, the molecular weight of polyethylene was around like 1,000 Dalton. Also, this vitamin E shows the antioxidant property, and this property is useful to reduce the reactive oxygen species that is produced during a dialysis process. And also the polyethylene glycol, which is a hydrophilic polymer, and it makes and that also is very good for making those membrane surface hydrophilic, so there will be less adsorption of the protein and the blood component. So, and another thing, like even though like some of the TPGs even diffuse in the blood, in case like it leach out from uh, uh, the hollow fiber, it will not create any adverse effect because its toxicity is really low, and 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 it is so that it is a benign material. So it can be the best choice to make this fiber biocompatible. So here what we did actually, we used we created like a four different or five different composition to make this hollow fiber membrane. The first one is a P is nothing but like a pure polymer system where there is no additives. So here like it consists of 25% polysulfone and 75% N-methyl pyrrolidine. Similarly, like PT5, it has 5% TPGS, vitamin E TPGS, PT10, 10%, PT15, 15%, and PT20, 20%. And we kept this all, we kept the polysulfone composition same and just varied uh, the TPGS composition. So this is the typical process parameter that we use to make a hollow fiber membrane. Like we try to keep the temperature same. That way, one can avoid the temperature impact. So we can make the membrane consistent. The humidity kept in between 50 to 60%. Then the temperature of the dope solution was also pretty consistent, like 25 degrees Celsius. And we try to keep the temperature same to avoid the temperature impact, so we can get every time when we make the material, when we make the fiber, it will produce the same result. And here we use this is the spin rate, like the inner diameter, the bore diameter was around zero point 0.8 and outer diameter around 1.4 mm. The air gap was like 45 centimeter. We use the RO water as for a coagulation bath and the rinse bath. The both tank temperature was around 25 degrees Celsius, and 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 the take up drum speed was 3.89 meter per minute. This is the speed like we kept constant, so it can also stretch some fiber, so some drawing can happen, and so you can make the fiber of desired dimension by 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 increasing the take up speed with respect to the extrusion speed. So this is the ACM micrograph of the hollow fiber, all five different type of hollow fiber that produced from the setup. And we try to you can see that this wall thickness of the hollow fiber like increases with increasing the additives, which is quite obvious because the percentage solid content is increased. And if you look at this is the closer view of this hollow fiber. This is the inside of the hollow fiber. This is the outside of the hollow fiber. In inside, you can see very compact structure, and it's very here you can see it's very difficult to like see where that where the porous structure start and dense structure like ends, and so for that reason we need to like have very high resolution image to know like how much the thickness of the selective layer, and actually this selective layer is responsible for the separation. This is the some of the structure of in the macro white, which is open cell type of structure, highly porous structure. And this is a surface. Actually you can see some some big bubble like pores, but if you have more closer view, it is highly porous surface. So can you hear me? Are you guys there?
Yes, yes, we are there. Don't worry. We are there.
Okay,
We are
okay.
here actually.
Okay, thank you. It's all silent, so that's the reason. I have to make sure when you
No,
are.
we actually see. This is not a common topic, so we want to learn from here.
Okay.
So we are, I mean, learning from here, and then we will ask some question answers.
Yeah, sure. Okay, thank you.
Yeah, welcome.
So it's very important to confirm the presence of that vitamin E in the polymer, that hollow fiber composite. So we conducted the first FTIR study, ATR-FTIR study, and from this study we found that there's been some new peak formation at these frequencies, at this wave number actually, and from this it confirmed that there is the presence of the TPGS. Also, the thermogravimetric analysis, the TJ, was conducted, and this is the first one is a pure polysulfone. And as you go further, like you can, this is the composite hollow fibers that prepared, and you can see there is this decomposition is not straightforward. And so the TPGS, like vitamin TPGS, is a low molecular weight material that decompose earlier, while the polysulfone decompose later. So based on that, we also confirm from this study that there is a 6 to 8% the TPGS content in the fiber, and it varies with it increases with the increasing the TPGS content in the actual polymer solution. So this is just a typical, like a schematic showing how that uh, uh, how that the TPGS is entrapped in in the the membrane. This is this like the green dots, like represent the non-solvent molecule, and the blue dot is represent the solvent. This is the polysulfone. The red lines, this the sorry, the black line shows the polysulfone, red shows the TPGS. This is this is the mixture of the polysulfone, TPGS, and solvent. When the phase separation happen, some of the TPGS that leach out from the polymer solution, some of the TPGS that get entrapped in the polysulfone solution, and this is the most of the cases. This phase separation, there is a phenomenon called vitrification. So there is a sudden freezing of the structure, and that sudden freezing of the structure helps to entrap the TPGS in the polymer matrix. and and in the phase separation at the skin consists the skin in the membrane skin. It consists of the nodules. Nodules is nothing but the aggregate of like a few polymer molecules actually, and these nodules are very tiny nodules. These nodules are connected to each other by entanglement, and the gap between these nodules is nothing but called a membrane pore, and that's how uh, uh, the selective layer of the membrane consists of. So this is the last one. This shows uh, the final structure of the membrane, how the TPGS is get entrapped in the polysulfone matrix. And this is this schematic. The purpose of this schematic is to show how the TPGS entrapment happen in polysulfone hollow fiber membrane. So further, like since this material being developed for hemodialysis application, it's very important to evaluate this material for a biocompatibility study. So what we did actually, we take these hollow fibers and we sterilize those hollow fiber, then then place those hollow fiber in the cell culture dishes and grow the cells on the hollow fiber. So if the cells are not growing, it means that there is something that is not good for a cellular environment. So here you can see that in all the fibers, like there is a nice growth of the cells on the surface of the hollow fiber, and this indicate that there is no these membranes have no cytotoxicity. So it clears the cytotoxicity, and it shows some compatibility with the cell the cell, and and it passes that test. This is a great result. The next what we did again, we try to like stain those cells, like like some better visualization how those cells looks like on the surface of the hollow fiber. So we stain those cells with the FITC phenolphthalein. That is the one kind of dye that that scale that stain the actin structure of the cell. And then the the blue dye we use DAPI, that stains the nucleus. So you can see like you can see the nice structure of the cells, and some and also the presence of the nucleus. So this indicates that these cells are like really healthy. They're growing. They are in they are and also like this cell growth is very profuse cell growth. And compared to, and also as you can see that this cell growth like density increases with the with increasing the vitamin E TPGS content, which is a quite encouraging result. Again, like we also culture the inner side of the hollow fiber membrane, but you can see here on the inner side, this is lumen side, and there is no attachment. There is no attachment of the cell Hydromer as such, which is really good. And this shows like it's serving our purpose, that it shows the minimal cellular adhesion, no cell growth, and it doesn't like support any attachment. So this is what needed for the dialysis membrane. The next week we also studied some of the reactive
I have one
Yep.
question here. Can I ask?
Yeah,
So
sure.
here, this membrane here, and would this body naturally would be enough to create that much pressure so that means some material can be fluxed out through in the membrane ball?
Actually,
How
what
much pressure does it need actually so that the something can can permeate through the membrane actually?
Yeah, actually, this mostly like this whatever the study, these studies are conducted in static environment. This is not in live, like this is
Okay.
just and but like in a,
Practically,
but in reality, when you have that hollow fiber cartridge, you need an external pump. They're called blood pump. It's a kind of peristaltic pump basically, and that pump used to like the purpose of that pump is to create the pressure to push that blood through the lumen of the fiber, because these lumens are very tiny, and that's how they created that pressure difference. So that way the material can diffuse from one lumen side to those toxin can diffuse from lumen side to the cell side.
so practically they need means the external pump actually during the dialysis
Yes, yes,
process, right?
yes, yes, they always use that one
Okay,
actually.
okay.
And and what they do actually, there is they use very high flow rate. In that case, the higher the flow rate, like more pressure like push through that, the more blood as you push through the lumen of the fiber, the better the separation performance, better the toxin uh, removal from the blood.
The same type of hollow fiber membrane are being used in case of let's
Yeah, yeah, yeah.
say
In actual
blockage of
actual
thing.
What you were saying, can you repeat the question?
Same type of hollow fiber membrane are being used just to take out the blockage of, let's say, veins in body.
No, they are different. Like what they don't, they doesn't say hollow fiber membrane for the for blockage or other purpose they use called catheter. It's a kind of like a metal tube or some polymeric tube actually. And in that case,
Okay.
that the tube no don't need to be porous. It is just a dense tube. Like it's a made from like a regular tube actually. So that's a different application. But this the application of this fiber is like this fiber is to remove the blood toxin, uh, uh,
Oh,
the toxin from the blood actually.
okay.
Okay, so
Thank you.
okay.
Yeah.
Yeah, it's something like a very interesting topic. I believe so. It might be interesting to you people, and if you're like whatever
Yes.
like details, they are really happy to provide
So
the answer to all the
happy and
questions.
yeah.
Okay, okay. And maybe later I can share this presentation with you if you people need. Okay.
Yes, yes. Yeah, continue.
Okay. Mm-hmm. So the next study like we conducted was a reactive oxygen species study and the plasma protein adsorption. In reactive oxygen species, we measure the fluorescence like there is a we use assay technique is ELISA assay technique and using like we take out the harvested the cells from the cells that attach uh, to the fiber surface and then using the ELISA assay, like we measure this reactive oxygen species. This is a very standard procedure. You can find the detailed procedure in the article that we published. So you can see that the the reactive oxygen species generation. We also use the commercial membrane just as a comparison. This Hemoflow F6 is the commercial membrane, and we also found quite like encouraging result. This one very exciting result where the here in this case lesser that reactive oxygen species the better the membrane. So you can see like we have done the cell culture study for like a 5 days, like 5 days, and as the time like this time increases, the ROS it's obvious that ROS reactive oxygen species uh, uh, content increases with the time. But with compared to like this first one is the pure polysulfone, the last one is the this last bar is the commercial membrane, and compared to these two, this composite membrane shows the lesser the reactive oxygen species generation. And then similarly, like we also like conducted the protein adsorption. We use the three proteins like albumin, globulin, and fibrinogen. These are the blood proteins, and we we made a we made this protein solution exactly similar to the content of those protein in the blood, and and incubate incubate these fibers with those protein solution for a while, and then and try to measure like then wash out those fiber, and then using the surfactant, whatever the protein adsorb, we take out that protein and then measured using the calorimetric technique, uh, uh, and from this what we can see that the protein adsorption significantly decreases with increasing the TPGS content, and so the TPGS is providing a good protein adsorption reduction, which is a good thing for the hollow fiber membrane. Similarly, what we did actually, we also take out we take the blood, separate the blood, RBCs or red blood cells and the plasma. And the plasma basically consists of the protein and the platelets. So we took this platelet solution, plasma platelet solution. We took this platelet solution and incubate those platelet on the hollow fiber, And based on like this is a pure polysulfone fiber. Here you can see there's a cluster of this platelet formation, and basically this indicates that uh, there is some sort of like a coagulation initiation. But like compared to other fibers like this other TPGS fiber, there's still like some platelet adsorption, but not that much compared to the native polysulfone membrane. And you can also see these platelets like they are like showing some tentacles. They deform. These are deformed platelets, but this deformation is not that much significant. And like in ideal situation, the platelet there should not any platelet adhesion or any deformation on any foreign surface. But this thing is quite significantly reduced with this the with the composite the membrane that we prepared. So also we also study this complement activation is a kind of like immune activation process. We also studied that like complement. These all these studies are conducted in vitro study. It's not in vivo. So this all these studies are conducted in static environment. That was the very first step that one has to do. We also like conducted some animal in vivo studies that I'll discuss later. So we also got like like this polysulfone TPGS composite membrane shows the good result compared to the native polysulfone and this commercial hollow fiber membrane. So this is the permeability of the hollow fiber membrane. This is water permeability. To measure the water permeability, and as you can see, the water permeability with the increases with increasing the TPGS content. This is quite obvious that as we have the more TPGS in the polymer solution, it diffuses out from the matrix and it creates more porosity in the polymer, and that's why like you can see more water permeability. And this is the rejection like plot, and like you might have heard about the molecular weight cutoff. That is very common common characterization of the hollow fiber membrane. This molecular weight cutoff is nothing but the molecular weight corresponding to 90% rejection. This is a percentage rejection, and if you draw the line for like from this 90% rejection all the way here, and whatever the molecular weight like corresponding to that point on this X line is nothing but the cutoff, the molecular weight cutoff of that membrane. And all these membranes like showing like uh, uh, very like comparable like cutoff is not that much significant increase, and which is uh, quite good. So this is one of the study like we conducted that is a this is a very high resolution SCM like high resolution scanning electron microscope images. This first image is at like 25,000 magnification, and the second image is 50,000 magnification. And as you can see that when you observe this, as I told you, uh, the selective layer of the membrane consists of the nodules. These nodules are like the selective, and this selective layer is closely packed nodular structure of the membrane, and the spaces between these nodules is nothing but called a pore or a pore length of the membrane. So you can see like here like with this is the pure polysulfone. Then this is the 5% TPGS, and as you can see that this pack structure pack the packness of the structure like decrease with increasing the content, and in this case this packed length is like the thickness of this dense structure decreased, and you can see more porosity here, like more high resolution like images. And these are the nodules that made from the polymer, that like it's very difficult to know how much like a TPGS in the nodules. We haven't done those studies, but we done ATR studies, and from that, like we confirm there's the presence of the TPGS. So this is a really a good result that we obtained. Then also we also done some like urea clearance study, like using this hollow fiber membrane, and and you can see that this urea clearance on the hollow fiber study is very similar. Like it shows very comparable to this commercial membrane. So indeed, this fiber can remove the, and also in this case, if you look at the diffuse urea per like a gram per like per area, like a square area, these fibers are shows much higher value than the commercial one. And this is quite exciting results. So conclusion of this, like the first part of the study, that we confirm over like the polymer that made from the hollow fiber that is made from polysulfone and TPGS composite shows the presence of the TPGS from, and as we confirm that is from ATR and TGS study. This also like this composite membrane also show the improved biocompatibility compared to the native polysulfone and the commercial membrane. So you have any questions on this, like in this section? Hello.
Actually, I want to ask the something actually because it's concerned to the membrane only. That
Yeah, but
let's
like
say
but like I'm like next one. I'm like in next section. I'm going to discuss of the coating actually, like the coating part. I know that this.
no. I'm not talking about coating. I'm just talking about the membrane. I have question to that only.
Okay.
So my question that you have hollow fiber membrane, you tested something. Let's say we have a membrane, flat membrane, just a polymeric film. We have created a membrane in the film. So
Okay.
that's just a flat.
Okay.
So suppose I want to measure its flux.
Yes.
So is there any technique available which can measure the flux of flat membrane?
Yeah, actually, what they call the cross flow filtration. Like you can make those cells. It's pretty easy. What they do actually, like I can send you some design actually. Like you can find those information, some books also. But uh, but I
So
don't know
there's a
what
low
our,
company which makes those type of cell, or we have to fabricate our own.
you can fabricate your own, and you can refer some research article. I can also send you those articles. But my question is that what type of those like films you're talking about? Is a dense, like it's a polymeric dense film, or it's a porous like a film, like, so it
No, no, dense
depend.
film so flux not calculate. No, because once it's a dense film, the flux is not possible. It is the porous film actually.
Okay.
So when we prepare a film, I mean, sometime we get phase separation, and that's become a porous type of thing. And we
Okay, I
want
see.
to check what the flux is of this particular membrane is. But those are flat in the nature actually.
Oh, I see. So what you can do basically, very first, I'll suggest you the very first characterization you can do is the scanning electron microscope of
That
those
we did.
films.
This is the course.
And if you see like, depends like what type of structure you have. Sometime like you can get a closed cell structure. If you get a closed cell structure, which is nothing but the kind of dense film actually, because there will be, it always, whatever the separation, it will happen to very diffusion. But when you get an open cellular structure where the cells are open, then it's a porous film. So based on that, you can decide what kind of equipment you have to use, whether you have to use the gas separation cell, or whether you have to use the water filtration or water permeation cell.
Okay, because I am in search of that instrument from several years, and because we want to test those membranes actually that we get in our in the
Okay, so
study room.
I will definitely help you to build or provide some design for those the permeation setup.
Okay, thank you very much.
Mm-hmm.
Any other
I,
participant they can ask in the meantime actually, because sometime what happened last due to some connectivity issues and so many problems, our question may not be answered in that time. So it's better to
yeah,
whenever you have a question, better that time only.
yeah, sure. I'll be happy to provide the answer to those questions. And
Yes, yes.
any
Okay,
questions?
continue.
Okay, good.
Yeah,
So
please,
the so,
if you want.
oh, what you're saying?
No, I think continue. No, no, you continue.
Okay. So next section, I'm going to talk about the in situ coating of the MPC polymer on a polysulfone hollow fiber membrane. Like this coating is performed on the lumen side of the hollow fiber, and it's not easy to coat the lumen of the hollow fiber. So the this like I the, so this MPC, it's a the name of that polymer is 2-methacryloyl oxyethyl phosphoryl chlorine. This is a biomimetic material that is found in the cell wall of the blood, and this is a phospholipid polymer. And we used this we source these polymers from like a commercial this is a commercial polymer we used, and these are like a PC2118 and PC1059. This is a the name or whatever the number of the name or the number that they have given to those polymer. So we use this polymer solution, and what we did actually, we use the same setup, but instead in the bore solution, we dissolve these polymers in ethanol, because these are water insoluble. This polymer is not soluble in water, so we use ethanol as a as a solvent for this MPC polymer, and also the ethanol can be a good coagulant for making hollow fiber. So as soon as the phase separation happen, this polymer also get diffused or adhere to the surface of the lumen side of the membrane, and we try to make this try to make this in situ coating of this MPC polymer on lumen side of the hollow fiber. So this is the process parameter we used, and here we make two type of the polymers. One is this is the name we have given, the PSF MPC1 and PSF MPC2, and these are two hollow fibers made from these two different materials, MPC materials. Uh, this is we also for a comparison purpose, we also made uh, the pure polysulfone hollow fiber membrane with the same composition. And as this is a typical microstructure usually we get from hollow fiber. We are similar to earlier studies. We also we conducted the FTIR of those fibers, the lumen side, and we found that there is some new peaks at the inner side of that inner side of the fiber. And this peak strength is very small because the presence of those materials really in very small percentage. So that's the reason actually the peak strength is small. We don't expect the strong presence. We don't need that much. So from this spectrum, it confirms that there is a presence of those MPC polymer on the lumen side of the fiber. So similar to the earlier studies, like we try to do some the biocompatibility studies. Here we can also see some cell growth on the fiber, which indicate that this fiber is not toxic to the cells. Similarly, we did a platelet studies, and we found the lesser platelet adhesion compared to this native fiber, which is pretty good. And then we also conducted some of the solute rejection study. And as you can see that the solute rejection, like a molecular weight cutoff, these fibers are very like there's not that much impact because of that new the material that we attach to the lumen side of the fiber. The performance, the porosity remains very similar to the native hollow fiber membrane, which is a good thing. So from this study, like we this was just the curiosity purpose, like we conducted this study. So how we can, like, coat this lumen side of the fiber. But and this polymer are like the polymer we used. One question you might have, that how this polymer get like this, the polymer we use, that is the cross-linkable polymer, as it get dry, as this polymer get dry, it cross-link itself, and this was like a self cross-linkable polymers, MPC polymer that we used. And uh, and uh, and those polymers formed, the presence of those polymers is not impacting the porosity as such, and it is showing the very similar porosity that of the pure polysulfone membrane. And these studies just conclude that, like, this is also the good way of increasing the biocompatibility of the membrane, where, like, actually mostly the blood contact happen on the lumen side. So it is very, like, critical to increase the biocompatibility of the lumen side. So this is the, like, the second part that uh, the study that we did actually. And the third one, like, this is another interesting study. Like, this is mostly like a lot of like experimental work we have done. And in the third one, like, in the third study, we did a in vivo evaluation of this material. We implant these fibers in the rat peritoneal cavity, and observe, like, how the tissue grows around the around the fiber. And based on that, uh, you can you can evaluate whether the material is biocompatible or not. And this is also one of the common technique the people use. And here, like, we have done like a 3-week study, and we take the sample, like we took the sample like a first week and third week. So so uh, this is just like a hollow fiber preparation. This is one of the, like, just photo that how we like implanting that the fiber, like, a bundles in the rat abdomen. We conducted this study at the Bombay Veterinary College with the help of like, and we have, like, taken all the permission, the animal body, like, a permission to conduct this experiment. So this is the weight observation uh, of those animals with the time. And as you can see that this is the basal weight and this is the final weight. Like, this is the weight at the time of implanting, and this is a final weight. There's there's the increase in the body weight. It means that this material is not, like, adversely impacting on the body. Similar result we found with the 21 days, like a 3-week study. So this all this result shows that the animal is growing healthy and no issue. And then after 21, this is a 7-day photo and this is 21 days photo. And as you can see, this fibers is a nicely covered with the tissue. There's also some blood cell or blood vessel formation, and 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 the, and this there is no any necrosis or any like a tissue damage. This fiber easily get along with the tissue. The and the new tissue are formed around the fiber. This is really a good thing to show that these fibers are indeed a biocompatible one. We also conducted some biochemistry study, like a blood hematology. This is like similarly we conduct the normal CBC count, and this and this from this result it shows like that the implanted rats were without any infection due to the surgery and implant. This is the normal CBC count. Similarly, we also did some serum protein analysis, so to check the kidney and liver function, and this result also show the normal function after the implantation. Then we also done some leukocyte analysis count to see that any inflammatory reactions happen. And so we take that peritoneal fluid around, like where the fluid that is around the implanted fiber, and we check with the normal, like a rat without surgery and with the surgery. It's not that much like significant difference in those counts. Those are quite comparable. These are some of the ACM images that we have done, and you can see there is a nice wrapping of the tissues around those fibers. Then we have done some histology study. Here we stain those fibers, the fiber with the tissue. We take that fiber with tissue, then slice those, then created using the slicing technique. We created the slices and then stain those slices with the hematotoxylin and eosin. These are the dyes they use, and this is the 7-day study, and this is the 21-day studies. And as you can see, uh, if you look at the closer, actually you cannot see from this picture as such, but if you zoom in, you can see there is some blood vessel formation around the tissue, and which shows some healthy tissue development around the fibers. This is also like we also use some of the another dye, like we use the Mason's trichome dye to section the tissue section. And here also we check with the collagen, like some of the collagen and other content, and what from this study shows that the thin collagen bundles were deposited for all the implant for in all implant for 7 days as well as 21 days. So we also did some immunochemistry analysis, and from this we got those samples, stained with those sample with the dye, and check the presence of those cells around this, just the confirmation. So conclusion from the studies is that all the CBC count and those things shows that linear and rival function is fine, and there is no side impact. from the peritoneal like fluid that we took from the around that is surrounded by the that is around the implant, shows that there was some initial inflammation with the implant, but it returns to normal at 21 days. The histopathology studies that we show that there is abundant fibroblast and angiogenesis. Angiogenesis is nothing but the formation of the blood capillaries, and this is a which indicate that that these composite hollow fibers are more compatible compared to this commercial and the native hollow fiber membrane, and there is no new immune response against like all the fibers implant. So I would like to acknowledge this work to my professor Jayesh Bellare, my PhD advisor committee Madhu Minjamur, Professor Anil Kumar, and my colleague Rohit Tiwatia, Amit Jaiswal, Dr. Sachin Kadam, and the members of the Silicate Lab that was like during the 2003 to 2011 time. And I would like to also thank you to the Department of Science and Technology for the funding this project. The SAFE, which is the analytical center for characterizing this fiber, and then also the Department of Metallurgy and Department of Physics for the utilization of their equipment for characterization of the materials. Yeah, that's all. Thank you.
Yeah, thank you very much, Dr. Ganpat, for sharing this. It's totally very much deep technical detail about hollow fiber membrane
Mm-hmm.
and their application directly. You are so this application side is being done by your company, or you just have some other partner to do that actually?
No, actually,
Is that
this was just academic study.
okay?
There a lot of things need to be done actually when you make the fiber. Like you have to do animal trial, human trial, all those things. You have to take the FDA approval because it's a quite lengthy process. It's not easy
Right, right.
to it's not easy to
Yes,
bring
to
any medical device to the market.
to
It
develop,
takes
I think the medical thing is takes practically a period of 10 years, I think, as per the
yes, yes,
normal protocol.
yes. It takes a long time. But like since this was like a good start, so a lot of like a PhD student they have worked on like a further on this hollow fiber stuff. And when I left, actually, like we filed the patent on this one, the composite hollow fiber membrane. We published like a lot of articles on hollow fiber fabric, like a hollow fiber membranes. So I don't know like what the status of like since currently I'm working in different area, but I don't know the status of the what the commercialization happened or not. But it will be happy if it is happen. I don't know. I'm not aware, but
What about your company
this is yeah.
at present? What about the your company thing? So they are also working in the same field?
No. Actually, what we do actually, uh, uh, we get all the medical devices, like all type of medical devices. Mostly, like we get the catheter, as you discussed, like as you're talking about heart blockage and all those things. So what those catheter need to like when you get those catheter, nothing but the tubes actually that just go through the blood vessel. Okay, and so what we do, we do the hydrophilic like lubricious coating on those catheter. The main purpose of those coating is that those catheters should cell like a cell like go smoothly through the capillaries through the blood vessels, and then it should not like have any adverse side effect, like it's not like having any reaction or anything with the blood. So that like we quote like a lot of devices like all like a cardiac catheter, some therapeutic catheter we do quote. We some urological catheter, abdominal catheter, some neurological, the bronchial. So some of the implant also we quote. So this coating is very like a this coating is a lubricious hydrophilic coating, and it will be great. Like since you work in the coating area a long time, this can be also interesting to you can also do some like start some research in this area to make some coating for these biomedical devices.
Yeah, you are the right, but for that we need probably much more high-end facilities. I hope so, right?
No, actually, mm, for those coating, like you can like what you have to do like have some frictional like a friction testing machine. You can have some friction testing machine. That's it. And also like there is some way like you can like see the coating is there or not. Like you can have like they use some dye test they called crystal violet, that the dye they use to like make sure that coating is there, like the coating, like one of the component of the coating, like the hydrophilic coating, the OH group that react with that crystal violet, and it stains that coating. So you have to do like you don't need to very high end facility, but by using some basic thing you can use. And if you want to do some like higher studies, you can send those
No,
sample.
actually, my concern is that let's say we want to test on some in vitro means applications.
uh
So in that case, it need a lot of approvals, right? In that particular time.
Actually, you can go with some commercial institute, or some you can tie up with some biological institute or some veterinary colleges, or you can tie up with them and you can get it done. I mean, there are also some the companies they can conduct for you. I
Okay,
think,
okay.
yes, yes. So this can be also a good study actually. I mean, it has a more practical application. So any other
Yeah,
questions from anyone?
yes, yes. But anyone else wants to ask something? Any participant wants to ask any question before Dr. Ganpat take the leave? Because it's you can say for him it would be around
Two. It's a
now.
two in the morning actually.
So yeah, yeah, 2 a.m. actually. So I am really actually obliged to you to actually manage the time at least and share your expertise here. Anyone wants to ask, they can ask so that we can close this session here at least. S- so if there is no questions, actually, I would say I will thank you very much. Actually, again, to be the part of that, managing your time
Hmm.
and sharing this I mean, information, which is applications of high-end applications of polymeric coating, actually. So in medical applications, and which is very crucial actually because in this means failures and toxicity, even if actually a small level of toxicity would be very dangerous actually. So means you should be very very specific in designing of such type of coatings or applications actually. So that need lot of cares. So but the research should be extended in that particular field actually. So thank you very much. So with this, I would say thank you once again to be the part of this short-term course. So if anyone wants to ask anything, we can ask, but otherwise we'll close this session here and we will reassemble at 1:50 for our the last session by Dr. Navneet Kumar, the head of Sales Europe Jindal Polyfilm, who would talk on barrier coating in food packaging applications.
So, okay,
Yeah,
Ganpat.
thank you for your time and
Yes,
patience. And if you have any questions, you can send me the email, or you can talk me offline. No problem. Thank
yeah,
you.
yeah,
Have a good one.
sure, sure, sure.
Thank you.
Yeah, yeah, okay.